It is. It is. Yeah.
Okay. So, first of all, welcome, Dr. Jailin Zhang. It's my pleasure to introduce you today. I was absolutely delighted when you accepted the invitation to speak at our seminar series a couple of months ago. For the group, Dr. Zhang is a internationally recognized scientist and academic leader who most recently has played a really pivotal role at Tongji University School of Medicine in Shanghai, where he's helped transform this School of Medicine into a globally competitive academic health science center. He's currently a distinguished professor of neurology and regenerative medicine there, as well as the dean and the director of the Brain and Spinal Cord Clinical Research Center. However, personally, I came to know Dr. Zhang through scientific collaborations prior to his move to China. And that was when he was at the University of Nebraska Medical Center, where I think you were there for over 20 years. It was when he was at Nebraska, we collaborated on studies examining the regulation of glutamate through inhibition of glutaminase inhibition as a therapeutic strategy for NeuroHIV and other neurodegenerative disorders. He even visited our lab in Baltimore when we were at Guilford Pharmaceuticals, I think back in 2003, so over 25 years ago. And I think he'll be interested to hear about the progress we've made since then on designing drugs to focus on inhibiting microglial glutaminase. Um, it At Nebraska, when he was at Nebraska, in addition to leading a very highly productive research program, Dr. Zhang held several senior leadership roles, including being the Associate Vice Chancellor for Academic Affairs and Associate Dean for Graduate Students. He's also a member of the European Academy of Arts and Science and has authored over 250 publications. As background, he earned his PhD in neurology in Shanghai and then completed most of his advanced research training here in the United States, including postdoctoral work in all our favorite areas, neuropharmacology, neuroimmunology, and in neurovirology. And his research has really been focused on the neuroinflammation and stem cell-based therapies for neurodegenerative diseases, and particularly our interest in this P30 seminar series in HIV-associated neurological disorders. And so super excited to have him today to join us, and please join me in welcoming him. He's going to speak on aging, microglial glutaminase, and neurodegenerative disorders. So welcome, Jailin. I'll turn it over to you.
Thank you very much, Bob, for your very kind invitation. And I also thank you very much for this very nice introductions about me. And I'm so glad to meet you online. And I was truly looking forward to meet you, your group in person. And I read and a lot of exciting work from your group and also your collaborators. And I'm just be happy to be here, and I'm looking forward to hear your feedback and critics to the work we've been doing. And clearly we are really today I'm covering three part in the background and also for the AD new inflammation, and also I'm talking a little bit about the PND, that which is very interesting we are working on because that can be done clinically as well. So we are really facing an aging society, and China has entered a deeper aging stage, which is by large scales and age-associated disease such as neurodegenerative disorders, and become a huge obstacle to human health. We are facing aging, different organs start aging differently, like the brain and the lungs start very early, and the skin and the muscle and in the kidney, they start different age, start of the aging process. And this age really associated with many mechanisms. It could be genetic, could be cellular, also the metabolic. Clearly, the chronic inflammation, oxidative stress, and the new endocrine, and also the epigenetics and also immunosystem imbalance play a very important role for this process of aging process. And for the mechanism, and we know that aging, brain aging could be neuronal damage, or the increase in your inflammation, or the abnormal protein depositions for it. For neurodegenerative disease, we actually know that Alzheimer's disease and also infections related and cognitive disorders. That's truly leading the clinical problem is dementia for that. So the others could be, many of the others we'll talk about later for it. And AD is clearly is the most common age-related, neurodegenerative disease and like a leading cause of dementia. The typical pathological things, including the A beta, plaque, and also the phosphorylated tau, tangles, and statistically indicate that annual cost of healthcare for AD is as high as more than 800 million billions. And for treatment for AD, there's not very much. And age over 65 in China, in total operation is already more than 12%. In Shanghai is even higher, more than 23%. So Shanghai is ranked the first in terms of aging, has entered a super aging society. And there is for AD clearly is high incidence. If it's over 65, there could be about 5%. If it's 80 years old, it could be 20%. So it's truly related, aging related. So although FDA recently approved like those A beta antibodies and most drugs developed based on the A beta or tau actually cannot have a very much of the therapeutic effect. We're looking forward in China doing a lot of clinical therapeutic usage of this and waiting to end of this year. Chinese neurologist society is going to give a summary how those antibodies worked in our clinical trials in that. Clearly, it is really essential to understanding the pathological of AD and dementia from a new perspective. And microglia-driven neuroinflammation, as we all know, a lot of people in the audience, and it's a key pathological features and also pathogenic factors of dementia. Neuroinflammation induces different many things, including the neurodegenerations and blood-brain barrier damage, and also demyelination as well. So microglia are truly responsible for A beta plaque, phosphorylated tau, or TREM2 dysregulation, or infection, or peripheral inflammation. So microglia is truly activations under the regulation of the inflammatory ligand receptor intracellular enzyme and also those transcriptive factors. So intracellular enzyme form a really good collecting part between the extracellular pathogen, and also intracellular response of a microglia. So, as a lot of people in the audience familiar with glutaminase is located in the memory of the mitochondria, which can hydrolyze glutamine into the glutamate, which is a key molecule for cellular energy and also for the metabolism. They can contain GLS1 and GLS2, and GLS1 is a dominant GLS member within the CNS of that. So GLS1, composed of two isoforms. One is a just one C-terminal called GAC. Another one is the KGA, which is a kidney type of GS1. And GS1 within the neurons which transfer the conversion of the glutamine to glutamate. The function of a molecular GS1 in microglia, which is still on a lot of research, and which is our group is really focusing on that. So, as Barbara mentioned, in many years ago when I was in Nebraska, Dr. Sean also in the group in the meeting today, Dr. Gandelman also, which is our chair of this. We are working on the beginning of that is HIV dementia, which is known as HIV associated, neurocognitive disorders. About 20% of adults and 50% children have that HIV, which is in the brain early stage, and the major cells in the brain, which is the microglia. So we've been focused on these things, how that the major cells in the brain, which is microglia, would involve in the interactions with astrocyte, with neuron, with blood-brain barrier damages, how that would be. But they invent cells are major cells in the brain, is macrophage, microglia. That work was in 1998 when Dr. Gandelman and actually we work on one of the HIV young patient. The patient is over 20 years old, and which is she did not know she got HIV infection. But under the heart therapy, including the ibuprofen, And after 1 month treatment, we can see an improvement in cognitive impairment, also in the MRI. Finally, the patient truly reversed from dementia. She went back to work as a kind of a teacher. Because we know microglia is major cells in the brain infected by HIV. If you with heart therapy get rid of virus, get also anti-inflammation, you can reverse a young patient from dementia. That's truly give us hope. If we target microglia, that might be the way to help with the disease such as dementia as well. So, clearly can detect that many of cytokine in the CSF. And lately in Shanghai, when we have COVID, we see we collect the 215 cases of COVID-19 and also 250 cases of a control group. We can see the cognitive function score of patient with COVID was significantly reduced. And we specifically see those patients over 50 years old have a serious cognitive dysfunctions. Female patients have a more severe cognitive dysfunction than the male patient. So in this way, and as we know, also COVID-19 is also a double-strand RNA virus as well. So clearly, the virus-related HIV dementia could be related to such as like a microglial inflammation in the brain, and also can cause those age people with cognitive impairment as well. So so then talk about what's the AD part of that. So GLS1 located in the mitochondria, they have a brain function, brain region of the GLS1, which is brain enriched, and GLS2 and the KGA due to the alternative splicing. What do we see earlier? We can see GLS1 is HOP activated in HIV dementia, and specifically in GAC is highly expressed in HIV dementia patient as well. The glutamate is accumulating HIV dementia as well. And lately, we determine the dementia of the patient. We see the glutamate in the serum as well for that. So lately, we did the 80 mice. We can see GLS1 is highly expressed in microglia of the 80 mice. And upregulation of the GLS1 expression is exclusive observed in microglia of 80 mice. Then we can see that GLS can promote the expression of the pro-inflammatory factors, and it can be overexpression of that. It can see more of the increase of those inflammatory factors too. So if we in the transgenic mice of GLS can induce cognitive decline, and that decline actually is using a nesting conjecture of the GAC in that process as well. So later on, we conditional knockout of GLS1 in microglia. We can see an improvement of that cognitive impairment of AD mice by the specifically conditional knockout of that. This data is not published yet. The following of those data is not published yet. I will go really quickly and just share with you, and we can see conditional knockout can inhibit the microglia activation and A beta accumulation, and that conditional knockout can reduce neural damage in AD mice. And the GLS inhibitor can also can see the improved cognitive performance of AD mice of that. And using the inhibitor of that, we can see that neuroinflammation was inhibited as well for that. We can see that can repress with CB839. You can see the repressed mitochondria damage. And we further detect if the GLS can interact with what kind of protein in that. We checked on that. We can see the GLS1 can mediate the interaction of the VDAC and the HKs. And that's by these different things we can see that interaction of those proteins. And that can see that using the HK1 flag and with the GHA we can see this interaction as well. So we can see that GLS1 can mediate the mitochondria localization of HKs, and GLS1 can modulate that metabolism of the microglia in this process. So clearly we can also see the GLS1 can control the mitochondrial memory potentials and the ETC proteins, and they can regulate the mitochondria mitophagy of that process in terms of mitochondrial fission and multitude for that. So we can also see that GLS can enhance the oxide stress of the microglia, and they can enhance the extracellular. If we overexpress the GAC, we can see the increase of the extracellular vesicle release from microglia as well. This one is published. So we can see the GLS inhibitor can block the EV release from microglia using the CB839 for that process. And we can see the GLS induced EV release is actually glutamine dependent of this process. And also interestingly, we can see the alpha kilo can actually, reverse the inhibition of the microglia by the peptide CB839, and the alpha kilo is involved in this process for that through the EV detections. We can see GLS can increase the inflammation potential of the microglia EV. Later on, we overexpress of GLS, we can see it increase of the pro-inflammatory microRNAs in the EV as well. So in this work, what we we can see the GLS condition knockout of the microglia. We can see the reverse of the behavior and also the pathological changes in this AD mice in this process. And this actually we feel if we truly tackling on microglial glutaminase, that could be a very important therapeutic potential for AD and for the process. So, you know, I'm give a little bit talk about this, PND in this way as well later for that, But before we talk about that, and I would mention some kind of effort in our clinical team and also our basic research team in terms of how we come after the AD with could be potential AD therapeutic strategy for this process. And clearly, we have a cohort of the AD cohort in our Tongji Hospital. We have the over 3,000 of MCI and AD and cohort. We established a community and a clinical cohort to see if we can help with the multimodal of the database, the biomarkers in this, including the blood serum markers and also including the CSF markers to help with kind of a early screening and diagnosis of cognitive impairment. We also developed the wearable EEG devices and brain function detection system to help some indicators of cognitive impairment of early screening and diagnosis for that. We actually have a kind of a design remote brain health assessment and for this mild cognitive impairment detections based on the smartwatch and smartphones, and to using the wearable EEG and the wearable things for this detection to see if that can help for the detection of the early incidents along with mild cognitive impairment for that. Then we work with a group of people in different part of China. We using different functional and imaging and CSF biomarkers for the brain aging. And in the same time, we just started about 2 years ago, started 7,000 of the people who is aged over 55 in our Pudong district. Now we have more than 7,000 patient cohort in these things for this brain aging process. See how the brain aging will leading into potential cognitive impairment. See if some of those biomarkers can see those multi-scale, multi-dimension changes in the brain during the aging process, and can be clinical testable for that process. So for the intervention strategy, and what we see is clearly, as I mentioned, GLS1 can be one of the most important part. I'm truly looking forward to discuss with Barbara and yours and to see those glutamine antagonists for that will be very important. Second things, axon mediated neuroinflammation and cognitive impairment that can be could be a potential target. We also work on the long brain axis. We are working on the pulmonary hypertension patient, over 1,000 patients. We look at the difference between the pulmonary hypertension with cognitive impairment with the patient as well, because we do have you know, pulmonary hospital in Shanghai. We have a very many of the pulmonary hypertension patient, over 1,000 patients we have collected in this trial to see if the new target strategy in terms of brain non-access for this process of that. For a lot of things, we can using exon injection of exon, using new stem cells exon for these novel things. Then we can also in situ injection of the catalytic particles, helping with the treatment of antioxidant using a pod, insulin or the RVG things for this administration of exon for this process. The second part of that, we try to identify this different biofluid and using this biomarkers helping testing, see if this can be helpful for see the improvement in our clinical cohort for that process. And using protein biomimic synthesis of the catalytic material, using somatic cell reprogram the new induced new stem cell EV for a process, and also using that new stem EV for the therapeutic strategy in our animal model study, and also we're in the process using a human IMPC for that process for the potential human trial for that. And clearly using designing of the new screening of the drugs using the organelle-based the drug version platform and see if the new drugs can be for the targeting we are testing for the process. And clearly, also the non-pharmaceutical approaches, which is we're using the PND already, and in which is for the if the cognitive training process, and for that for the non-pharmacological approaches in the testing of this, and clearly that with the AI plus the BCI based technology, which is a testing by this different testing in the group for this thing, see if that can be helpful for the therapeutic potential as well. So what do we believe that because AD is a very challenge disease, we feel like what we see over almost 30 years ago now, and we can see the heart therapy can be good for HIV dementia. And what we can hope if we can using multiple approaches for AD for that could be combination of the blood flow improvement, which is we see drugs testing in our pulmonary hypertension patient to helping with also helping with brain blood flow improvement for that in combination of the development of the such as like neuroinflammatory inhibition, like glutaminase inhibitor with kind of a stem cell derived and the treatment for that. And those things in combinations can help with the peripheral and the brain interactions of improvement for that process. Clearly, those pathogenic factor in relation with A beta things of that, or in terms of a new strategy in neuromodulation with TMS or DBS for this, or with the new technology with the BCI-based cognitive training of that. So we hope with one or two or three of those combinations, we would come with a hope for the treatment of dementia, which we don't have any good drugs right now for the therapy for it. So, inclusion of the brain enhancement truly into the Chinese national health strategy and application of the brain enhancement of the multidimensional with from the passive treatment to the active prevention to explore interdisciplinary integrations and the clinical translation of this new MAD strategy in our basic and translational and also some of those cohort studies for that. So we do have a innovation center for basic research in the brain aging and association, and also critically with interaction with our Shanghai anesthesiology and the brain function labs with together with our Tongji Brain Center. We are working on this goal for that process. I'd like to mention to Barbara and also colleagues that we do have a brain aging, glial, and a neurodegenerative disease. We have been doing this in the past 3 years. This year's will be, in November, 6 to 8 in Shanghai. And I have invited Michael Priest and, a few colleagues. They agreed to come over. And I'd like to also extend my warm welcome to Barbara if you can come to China, Shanghai, this coming November. And I hope after Trump visit in April, and since maybe tension maybe release a little bit for that. And this, I believe, this meeting will be helpful promoting the interactions, collaboration, discussions among groups in different part of the world for that process. So for that part, we do have the internal mechanism, how that will be in terms of microglia. An external mechanism could be through different part of that infection or interactions or potential intervention for the process. We believe that MAT can be the new hope for dementia treatment for that. I'm going to spend a a few minutes about the PND, which is that's a very interesting part. I do believe the PND is a really important human can be done clinically. How that will be cognitive impairment can be improved by different means in clinical for that process, and that is truly of the how that associated with operation with anesthesiology relate to the neurocognitive disorders, which is have the cognitive impairment, and a brief at a PND for that. The PND is a condition characterized by the cognitive impairment affect older people after surgery and anesthesia, and actually, that can be age related, and there is and many different things can be related, and also the anesthesiology. And if we have the age higher, when older than 65, and they can have a high incidence of a rate, they would double of the, for that. For the heart cardiac surgery, even the incidence can be about a quarter for that. It's a severe disease and a burden, and increase of AD risk about more than tenfold of that. Incidence of this post-operative complication as well. So there is a cognitive dysfunction patient during the perioperative period, and you can see the AD and the PND, and there is an epidemiology relationship with aging group, and also surgery with aging group can have a high incidence of the PND as well. There is a clinical magnification for that. In terms of potential mechanism, clearly neuroinflammation can also be important in this process as well. So the PND can be degeneration of the many of similar of the clearly new information can be a key pathological factors as well. The microglial exercise are activating PND, so the specific mechanism also remain under investigation in this process. So we did see of the GS1 level also increasing our PND microglia, and also the can promote the axon release, inflammatory trigger the microglia activation. And also we look at another pathway briefed as LCN2, which is the key markers of the excuse me, of the reactive exercise in promoting the inflammatory response and the LCN2 level increase the PND and the lockdown of the exercise LCN2 can rescue the cognitive dysfunction PND and MERK-TK inhibitor can also help with the PND as well for that. So GS1 clearly, we talk about GS1 release and trigger the market activation, how that relate to the PND process as well. So for that, we first with you know see how that help with the kind of a biomarkers associated with PND because they do have a diagnosis challenges of the neuropsychiatry skill with the brain images, with EEG, and also with the biological fluid biomarkers of that in the process. So lack of the objective and unified diagnosis and predictive of the PND, and also clinical predictive lack of the effective diagnosis, prevention, and treatment for the PND. So to discover PND biomarker with high sensitivity and specificity, so our group and Dr. Liu, which is the chair of anesthesiology of the Tongji Hospital, with over 25 of the chair of the anesthesiology and with neurology people together, we come up with a kind of a biomarkers, imaging markers, EEG biomarkers, and molecular biomarkers for the PND detections. And we are just for the first phase, just published in the last year for that. We're looking forward to do more of a cohort study in this process, have a large scale of clinical cohort-based construction of this prediction of that, and to see if that can be helpful for this different dimension prediction model for the PND, which is that's really associated with aging and with operation related for that process. And we just have a paper recently accepted with the TMS based of the PND interventions of using the TMS treatment to improve the patient short-term memory abilities after surgery, and they can inhibit the extensive activation macula in the mice, and also they reduce the mitochondria fission in mice of that process. So this clearly with the TMS can be beneficial for this process. And clearly the other means of the music therapies and the others can be helpful as well for that. So for that part, I just give a very brief introduction of the PND, which can be multi-dimensional prediction model for that, and also for the related with interventions in terms of the model, animal model studies, in terms of magnetic study, still undergoing in the lab and in our group for that process. So I'd like to really thank the people in my group since I returned to China as a full-time, faculties in about 9 years ago, and which is October of 2016. And I do have my Professor Xia Xiaohuan, who actually with Zhu Bing and Wang Yi and Xi Xingrui, and those are the faculty member, Zhang Xiaoming and Gao Ge, they are the faculty member in our group. And I also do have a postdoc fellow and Li Yun, and which is have one of the Nature Aging paper, in terms of mitochondria relationship with aging and neurodegeneration of that, and those graduate student, and our clinical team and chair of the neurology, Li Yuanxia, and also with postdoc fellow and junior faculty Li Cui and Zheng Xiaoran, and they are working on the cohort of this study with our MCI patient, and also with aging group in Pudong district for that. With our anesthesiology department, the chair of anesthesiology, chair of department of the Liu Jinghui, who was my postdoc in Nebraska. Now she's a chair for anesthesiology department. She's heading a team specifically with a group of the scientists in anesthesiology basic research for this process. So I do feel the basic and clinical research together with neurology and anesthesiology help with the work and with potential mechanism investigation the target for therapy, and also the new area for the PND, that could be something helping with our AD research as well. So I'd like to stop here to take any question you may have, and if I cannot address question, I will go after you go after Barbara because it's 1:40 a.m. in China right now. So thank you very much for your attention.
Very, very, very nice, Jai-Lin, for that excellent presentation and run through a tremendous amount of data with GLS and with Alzheimer's and in the PND. Especially given that it's 1:30 in the morning. I'm sure I have lots of questions, but I'd like to open it up to folks on the call to ask questions. I see, Jale, you've already unmuted, so I'll start with you. And then
Okay.
next, Marcus, you could go after Jale.
Yeah. Thank you, Dr. Zhang, for the wonderful talk. Really learned a lot. Yeah. So I know you also worked a lot of the disease conditions for the role of microglial glutamase. So my first question is, what do you think is the key function role of microglial glutaminase in the context of the AD, PND, or even the chronic stress condition? Are there common factors? Are there common pathways which you would highlight? This is my first question. Second question is I would like to learn your valuable opinion on what is the role of microglial glutaminase in mediating the microglial neuron interaction. Yeah, that's my two questions. Thank you.
This is actually two very important questions, and um, through the many years of study, and I truly come to more of a belief that microglia glutaminase are a bit, besides they have the function of transfer glutamine to glutamate, clearly there is a additional function of glutaminase, which is more of the GAC in microglia than just to transfer glutamine to glutamate. Right now, the inhibitor are more focused on transfer the glutamine to glutamate. That inhibitor actually worked in animal models in AD, in depression, and also in HIV model as well for that. We are still actually try to figure out what is the other function of GAC in microglia. I don't have a clear answer for that things yet. So I would also like to hear your opinions and also Barbara's opinion in this process. So if I can say for your first question, one for that. But clearly the inflammatory response driven by the GAC glutaminase is very important. It's actually is facilitate that pro-inflammatory acidicate of this microglia in the brain. But at the beginning that might be this is good at the beginning. At the beginning that might be good, but during the chronic process, even in our animal model study in the AD model and also in depression model, and I believe your group is doing the similar things too. That that kind of a chronic inflammatory response process being initiated or be continued by the glutaminase could be bad for this process. That we'll see from the EV studies from our, some of this work is under review right now, our mitochondrial EV studies as well for that process. So I think that could be things we need to still aware, need to be more investigation in this process. In terms of your second question, it's also if we look at the animal model work with our study, I'm sure with your study with your glutamine antagonist with CB839 of that as well. And this inhibitor is working for that. And also our condition knockout of mice is working as well. So that's further confirm our hypothesis if we balance that the chronic inflammation response driven by the glutaminase, that might be beneficial for this process in microglia. So if I can say that, I'm not sure. We're still working on that. I still don't know what is the exact mechanism for that process. Yeah. I'm not sure if I addressed the question, Dr. Zhu. Yeah.
Thank you.
Marcus, you want to go next?
Oh, yeah, thanks. Hi, Jolene. It's great to see you after quite some
Hey,
time.
Marcus, how are you? Oh, long time. Yeah.
Well, this
Yeah.
was a real tour de force, through your research program. I mean, it's fantastic to see. I mean, you can, address a lot of questions now at so many levels, right? And I mean, really between the model systems and the and real life. I'm also intrigued that you come upon lipocalin because we have been studying lipocalin too, in the context of HIV, and I think there is still it's really interesting to see because I think it's like many of the factors, and this leads to the question that I have, and is that lipocalin is somewhat at a crossroads, right? You can have we can see it can have some protective function because it seems to come up also as a factor that limits inflammatory responses, right? And this is you see, and it's very nicely fits with what you see. People have observed it post-operatively in, for example, in people, but also they have seen it in upregulated in people who have had a cardiac event and
Mhm.
is linked to depression, right?
Yeah.
So there is clearly this link to the talk or the interaction between the periphery and the central nervous system. And coming back to your glutaminase, because this is really fascinating. If you think about that, neurotransmission in general, and also in particular glutamatergic neurotransmission, is so tight, needs to be so tightly controlled, right?
Mhm.
And then and this is like a long-standing question that I ask a lot of the people who work on this is the we have, and you remember this, there's a lot of discussion that excitotoxicity plays a role, right? Was one
Mhm.
of the
Mhm.
mechanisms. And this
Mhm.
is why you think, controlling glutamate levels might be a critical component in limiting this, right? On
Mhm.
the other hand, if you look in neurodegenerative, diseases or impairment, people don't have too much glutamate. They have usually too little glutamate, right?
Mhm.
So, and that indicates, goes back to, so why is that sounds like, a paradox almost, right? But I think it points to the point. So I was wondering, when you have glutaminase activity, so what actually happens to the glutamate? Where does it go in the cells, right? And where do you see this activity? Is it all, in microglia, you would wonder, is it cytosolic? Because we know in neurons, everything gets, sequestered to vesicles. How does it work? If you think about microglia, how they regulate, is this just basically that they increase, basically, in principle, increase the pool of glutamate, which could be, if you think about the situation where there isn't enough glutamate, right? Might
Mhm.
that be a beneficial effect? Or, as long as it doesn't, overactivate the cells, right? And basically gets out of control. And because that then feeds back to the, where does glutamate act? And it acts at, of course, on the excitatory pathways, but it also acts on some of the inhibitory pathways, right?
Mhm.
So I was just wondering what you think, how this fits. Where is the glutamate going, and what what is the idea about its regulatory function?
This is it's a tough question. It's really a tough
Sorry.
question. It's a tough question. So because at the beginning, we all believe, or at least we believe glutamate play a very important roles in the process. We actually detect in our in vitro model, in our HIV model of that, we can see a very good of increase of glutamate in HIV infection macrophages. And also we see the inhibitor when that's how we started working with Takesh and using their inhibitor for our process, we see what we measure in the process is measure the glutamate accumulation in our in vitro model for that. So I still believe this is the excess glutamate would play some roles in the process because as we know that the mimicking, which is NMDA receptor loss inhibitor in the AD disease model and also in the AD patient, they're still working with early stage of the AD patient as well for that. So, but the more and more we work on the glutaminase in microglia and macrophage, what do we see that the increase of the microglia in the protein level is not the KGA. It is in more in GAC. That GAC, we try to uh, make the crystal structure of a GAC. We tried very hard of that because they have a very short form. I spent a lot of money on that too in China on that, but we still cannot the crystal structure of the GAC very well on that.
Mhm.
So we try to see if the GAC would have the more than different functions than the glutamine to glutamate functions in the process. We see an increase in inflammation. We see an increase in inflammatory factor. We see a lot of EV increase. But at this stage, we still cannot see their new function. There is one group by the Xiamen University's Chen Xiaofeng. They are trying to see there's a glutaminase new function in microglia as well. They are still also working on that as well for that process. Let me. Your question is, I would say, it does the glutamate play a role? I would say they still play a role, but do they play the most of the role, all of the role? I still doubt about it. So we are still trying to
Mhm.
figure that out. But that is a very, that is a very important of that. Hopefully, we can finally figure that out for that. What's the other function of the GAC in microglia for the process? Yeah.
Mhm. Okay, we will stay tuned for updates.
Yeah, but that's a very good question. It's so nice to see you after so many years. So great. Yeah.
See you. Yeah.
Good,
Thank
good.
you.
So welcome to Shanghai. Come to Shanghai and we can have more discussions. Yeah.
Takashi, you want to go next? I think your hand is
Yes,
raised.
hi, Jaren. Really
Hey.
nice talk. I'm really excited to see that you are really making a wonderful transition to the trans-regional research.
Yeah.
This is very exciting. So, question I have: I saw in one of your slides that the changes in the metabolism landscape upon activation of microglia, where you have more glycolysis and less oxidative phosphorylation, and that
Hmm.
reminds me of exactly something happening in the cancer cells.
Hmm.
And because of that, for the cancer cells, they have other targets beyond the glutaminase, especially on the process of glycolysis and other mitochondrial enzymes. Can you also see the possibility of other therapeutic targets that could be able to deal with this microglia activation besides glutaminase?
Takeshi, you
Mhm.
you you ask a good question. You answer a good question.
No.
So so um I still, interesting work is people is being working on the contaminants is one is in AD or depression in microglia. The other is also work on the cancer too.
Yeah. Mm.
So I do believe this is actually there will be a good connection there as well for it.
Right.
Yeah. So this is clearly this is beyond besides the contaminants. There is also the others in there as well too.
Okay. All right. Thank you. Mhm.
But this is very good, and it's um, you took me to the inhibitor of glutamate, and you remember you send all those inhibitor with you know you only know those code, and after we release the code, and you release
Yeah.
the code, we can see exciting inhibition of that, right? So
Yeah, so in general, we like inhibitors.
yeah,
So just wondering if there any other targets that could be useful in this, especially in the glycolysis, because some
yeah,
of those pathway we may already have a good inhibitors, but we
that'll
can
be
have
good.
some more talk on the offline. Yeah,
That'll be
nice
good.
talk. Thank you.
That's great.
Mhm.
Thank you, Takesh. Thank you. Yeah.
Jonathan, I had a question. You went through very quickly the data on the GLS conditional knockouts in the microglia, and you
Hmm.
compared that to the efficacy you got with the CB839. But
Hmm.
you went through it so fast, I couldn't I couldn't get the end result. Did you see similar efficacy with the knockout and the drug? And the reason I ask this too is those drugs are really lousy brain penetrant.
Sure.
When
Yeah.
we do brain to plasma ratio, they're quite weak. And so, did you measure how much of the CB839 got into the brain, and was it sufficient to target engage?
Sure.
So, anyway, a comparison between the knockout and the drug, and then also, what do you know about the drug exposure
Mhm.
necessary to see the efficacy you observed?
Yeah. Very good question, Barbara. We see better of the condition lockout clearly is much better than the drugs for sure. That's true. That's a young answer for that. And also, for the drugs, and I saw that the penetration of the blood-brain barrier is not very good, and but we surprisingly see it worked, but it's not as a good efficacy as compared to the condition lockout of that as well. So that to simply address your question for that process, yeah.
Did you have to give higher doses than was used for cancer? Then, given the fact that you get so low brain penetration, was your doses high?
With the cancer part of that, and clearly, the cancer part of that is they need to be good concentration for the cancer treatment as the people do that. And that drugs, I need to ask Takeshi for that too. That drugs in the company for that produce that drug for the cancer clinical trials stopped in the stage 2, right?
Yeah, they went bankrupt. Yeah, Calithera.
Yeah. I know what's going on with the drugs, and any people picked up on that afterwards or not?
Calithera was completely focused on an oncology, and the data was very modest.
Hmm.
They had better data when they combined with other oncology treatments, but as a single agent and even in combination, the data was just not that robust. And so I think investors backed out and the company has folded.
Hmm.
So I'm not aware of anything continuing with that in oncology. There is, and you already pointed this out, several people that are looking at the same class of compounds for AD and for depression. But again, the difficulty from a drug perspective is they just don't penetrate the brain very well, and so you have to give high doses to get enough there. Um,
Barbara,
so yeah.
seems very interesting. I'm not sure why, and in the AD mice, even the drugs, the penetration brain is not very good, but in the AD mice, we can see a good significant decrease of those brain formation and cognitive impairment.
Mm,
And this is very surprising us about, but it's not as high efficacy as compared to the condition knockout for that. But we do see that. I
do
don't know
they?
why, but that's why I like to collaborate with you with your new conjugated inhibitor to see to compare that. It'll be good for that, clearly.
Yeah. And I think to Takashi's point about asking about other things that could be affecting the microglia, in addition to glutaminase that might be beneficial, I
Mhm.
continue to be struck by the fact that when we use selective GLS-1 inhibitors,
Uh-huh.
they're not as effective as when
Mm.
we use more global and broader glutamine antagonists. They weren't as effective in cancer, and they're not as effective in brain. So there definitely are other activities that I think are contributing or beneficial.
Yes.
Uh,
Yes. That's what I believe too. We feel why because I did have about 2 to 3 years being stuck in macrophage and the regulation of glutaminase in HIV and inflammation-driven upregulation of that we finally realized it's not the KGA, but it was GAC. That was actually
Yeah.
the guy actually helped me in Colorado, and he now he retired now. He's been very helpful for that process. Yeah.
Yeah. Well, this was wonderful, Eileen. I'm just looking, we're coming to the hour. And so I just want to thank you so much
Yeah.
for participating today. And I know everyone really enjoyed that presentation. So thank you. Thank you very much.
Thank you very much,
Um,
Barbara.
Thank you,
And
Geraldine. Nice
and
thank you
talk.
very
then
much. And
before we go, Caroline, can you
Oh,
say what's up next week?
So there's
We're at
no
Croy.
seminar because it's Croy.
Oh,
But
okay. Got it.
then, so the next one is on March 4th, and it's Marcus Call.
Oh, well, excellent. All right.
oh,
Excellent. Very
Michael, so I'm
much.
looking forward to hear the Michael next time.
All right. Yeah. And Jalen, if you'd like to get on our list, although
Yeah,
in general it'll be 1 a.m. at your time to
that's
contribute,
okay.
we can certainly send you and maybe Caroline, you could send him the link to be able to join.
Please
But
send
thank
it.
you very
That'd be
much.
great.
Really,
They're great.
yeah,
Thank you very
really,
much.
really
Thank you very much.
wonderful
I'm so
today.
happy
Thank you.
thank
to
you,
connect with a
Geraldine.
lot of people. Yeah, yeah.
Yep.
So
Yeah.
Bye-bye.
bye bye. Thank you very
Bye-bye.
much.
Thank
Bye
you.
bye.
Thank
Bye
you,
bye.
Dr.
Bye-bye.
John.
Thank you very much. Dr.